Thank you. Thank you again for having me. So I'm Dr. Bjorn Oscarsson. I'm at Mayo Clinic, Jacksonville. And I am pleased to be invited to give this update to you all about this project that we have been running on the drug called Ibudilast. And again, living with ALS or caring for people with ALS, it's still a hunt for better and more effective treatments. And this is one of the many projects that currently are underway. One that I think is exciting in many regards, and maybe particularly exciting in some. So it is a project that, again, I'll share with you here first a bit about what we're doing, and then we'll have a dialogue about, well, I try to answer some questions regarding it. And again, hopefully you'll come away with more knowledge in regards to the product. I'll cover a little bit of background, the trial we're doing, and summarize sort of where we can go from there. This is a product that is already approved in Japan. It's been around since 1989 there with an approval. So the indications in Japan are treatment of asthma, breathing difficulties, and dizziness and other difficult symptoms after stroke. So not particularly dangerous diseases necessarily, not diseases where one would have a high tolerance for side effects or issues, but conditions where we have other, at least for asthma, effective treatments. The safety profile is therefore very well known with the product. We don't really expect big surprises as we're doing this study in ALS. The doses that are generally used in Japan for these symptoms are lower than what we're studying in ALS. We're going up to 100 milligrams here, as we'll touch on later, but that the doses usually used in Japan, the 20 milligrams a day, it is very well tolerated. Eventually, if we push the dose up higher, there seems to be a little bit of GI side effects, and that's kind of what's been seen in other trials using this product, but not any dangerous side effects. Again, these are tolerability type issues. So what does Ibudilast do? There are several mechanisms that are proposed to on how the product may be effective in helping a neurodegenerative disease like ALS. It is thought of as an anti-inflammatory drug in many ways, and it does this by inhibiting this macrophage inhibitory factor. It is also a phosphodiesterase inhibitor, and it has an effect on toll-like receptor 4. In addition to that, it also has a autophagy enhancing function that's less well studied but been shown in some work. So these kind of anti-inflammatory effects that have been seen with the drug in different model systems include an effect on interleukins and other pro-inflammatory markers, as well as upregulations and some growth factors. So again, it's not a single effect from this drug, it looks like, or at least there are several mechanisms that may be at play. Again, when we think about ALS, our understanding is somewhat limited, and there is a lot of interest in dual target engagement or trying to hit several targets at once in ALS. And again, the drugs we already have are striking in different places. So this kind of multimodal, more dirty approach may be important. At least it is not out of question that it is what's necessary. And here is sort of a capture of some of these studies that again point to the effects that I had on the earlier slides. Again, it's a smattering of studies, and again looking at different aspects of the drug. The product has been tried in other neurodegenerative, neuroinflammatory diseases. Again, we don't think of ALS as a primary neuroinflammatory disorder, but there is the inflammation part of as part of ALS, and that's something that's well recognized. Some work is very much focused on that, whereas other studies are more leading into the degenerative process that probably is the dominant aspect of ALS. Primary progressive MS, on the other hand, we're kind of in opposite situation there, where we're thinking of it more as neuroinflammatory and less as neurodegenerative, but likewise, a bit of both. And this is a drug that has been studied in this larger sprint study, which did show some positive benefits in secondary progressive MS. I touched on this autophagy enhancing aspect of the product. Again, not a tremendous amount of evidence for this, but it has also been suggested and could likewise be one mechanism that's important in ALS. So thinking about ALS and the multiple types of pathologies that we see, we could see that Ibudilast has an effect on several places, both on autophagy and proteostasis, again, as mentioned. That is one of the more central problems in ALS from our current perspective. And so any effect there could be very valuable, even if it is minor. There is evidence for reduced excitotoxicity with the product. This, again, is the mechanism behind riluzole. Its importance in ALS is also maybe more not central, but still valuable to interact with, as proven by riluzole. And the inflammatory portion there didn't show up, but yes, inflammation, again, being one of the features that we do see in ALS, which we still a bit struggle with interacting with. On to the COMBAT study. So this is the phase 2/3 study that we're doing. And this is a longer study. This is a study that I think is well positioned to answer the question quite firmly if the drug works or not. We're having a short leading phase with screening, really screening phase, not leading phase. And then we move into the treatment phase, which is a year long. This is not one of these shorter studies which are popular today. Many studies attempt to answer the question if the drug works in just 6 months, and it's hard to say. And I think we end up with a lot of products where we don't really fully, not everybody's convinced about an effect in 6 months. I mean, if we had a fantastically effective drug, I think everybody would be, but most things that we are testing is it's difficult to see an effect. Maybe on neurofilament light or other serum markers, we could see something in such a short time. But for most classical studies, including this one, which we now have been working with for years, which are powered on ALS Functional Rating Scale and survival, we need a longer study to be able to achieve a reasonable degree of certainty. This study has an open label extension phase afterwards. It is relatively short. Again, it is something, at least, that I wish that we had been able to make a longer open label extension phase, because we do have people who are completing the blinded and open label extension phase who want to keep going. And again, that's our current limit there as what's given by the study. The study itself, we have a screening visit, baseline, 6 weekend, and 3 months, 6 months, 9 months, 12 months, 15, 18 months. So we have a few visits early in the study, but then can fall into a every 3-month visit, which is pretty consistent with our typical care schedule for people with ALS. So this is a protocol that fits with what we're already doing to our patients. So it is intended to be it's always burdensome to be a trial. In a trial, it is a lot of effort from anybody who's willing to volunteer, but we're trying to kind of at least put the windows in a way that makes that as easy as it can. I already mentioned our endpoints. Again, what we're looking at is a change in ALS Functional Rating Scale and death and survival time. So it's a combined endpoint that we're looking at. We're also looking at the change in muscle strength, quality of life, and the functional activities. We have a responder analysis, and we'll kind of come back to why we have that. The Phase 2A study did suggest a responder group. And again, that is something which is one of the more intriguing pieces that we have. I think, in truth, much more exciting than the preclinical evidence. Again, the clinical evidence we have in ALS is really the most exciting part, and I'm saving that for last to kind of come away from this talk with some excitement. Death or time to permanent ventilation, we're looking at safety and tolerability, and relationships with plasma levels and the AEs. Key eligibility, we have a pretty broad range here. We can accept patients up to 80 years of age. Again, it's not a particularly difficult to tolerate product. People with ALS is who we're looking at. We do need people with early disease since we're powering on FRS. And having a solid ALS FRS score to begin with so that there is room to lose points. We think that's how we're going to get the most out of that scale, so we can answer the question if the drug works or not in the best way. Likewise, we're starting with a high breathing function. This is a product that it's a capsule. It's a small capsule. It's a size 4 capsule. It's very tiny, but people do need to be able to take them by mouth. We start at a dose of 30 milligrams twice a day for the first 2 weeks. Then we move up to a dose of 50 milligrams twice per day. And the reason for this escalation is these potential for GI side effects. It is a study where we also have an allowance where we can drop people down. We can also adjust the dosing schedule. So we have not had a lot of problems with this, but it is something where, again, we're pushing the dose quite a bit above what it's been used at in Japan. So this list is not fully up to speed, but this is roughly where the sites are. I apologize for not having an updated slide on this. So yes, what makes me most excited about this product? It's this ALS 1201 study. This is where I get excited. So this was a single center study done by Dr. Ben Brooks at the Carolinas ALS Center. And this is not published data, but it's been presented. And again, this was first out in 2018. It's a smaller study. He had 51 patients randomized, 2-to-1 on drug and versus placebo. And the study was negative overall, but there did seem to be responders, and there were more responders in the treatment group. So by this definition of responders, which is the company's definition, I think the pre-specified one, we're seeing about 21% responders on the FRS, 51% on the ALS Q5, which at least while not as commonly utilized, or is definitely a solid measure, and also a greater effect on muscle strength. These are bigger numbers than what we see on the placebo group. There were a group, again, these are the responders that were seen in the treatment group and in the placebo groups. There were also people who did better over time in the placebo group. Again, that is not so rare, it turns out. It seems a bit counterintuitive, but I think it reflects the limitations of the function rating scale and then the high variability of ALS. Looking at survival, we have the patients in red completing per protocol, and then we have, so red is treated and blue is placebo, and we're seeing a longer survival period. Again, these are longer than the trial, so we're again extending out past the trial. And again, this type of post-trial data is always a bit questionable, but this looks interesting. Let's see here. So this one has our treated group as the zeros and the placebo group here, where we're seeing that at this cut point, half of patients who had received drug were still alive, whereas only 25% of the placebo group were alive at this cut point, which I think is a year, this one. So there was this, what appears to be a group of patients who held steady during the blinded phase of the study. And then we have a post hoc survival benefit in the protocol. Now, ALS is highly variable, and it's a small study. So does this actually mean something or not? Well, we're working to find out. But it is the variability of ALS is high. Some people have rapid disease, other people have slow disease. How much of that is thanks to anything that we do? It's always a discussion, and again, we need to keep an eye on our statistics to kind of answer that question. So wrapping this up, this is kind of the characteristics that we're looking for: people with early disease, with a fairly steep but not too steep slope on their function rating scale, with a strong breathing function, and we do have an open label phase. Now, one question that I expect I'll be asked, and so I'll kind of get into it, is sort of off-label use of this product, because this is a product that is commercially available in Japan. I don't recommend it at this point, but I think there are far worse choices for self-experimentation and off-label use. But I think from my standpoint, my perspective on products in ALS, when we're repurposing, or for that matter, really developing a new product, is our track record isn't that we go from success to success, but rather most things that we try fail. It doesn't mean that we should not keep trying, and that this is, I mean, it's important that we keep moving and testing, but the base rate, the likelihood of anything that we think theoretically should have a decent chance, I hesitate. And I think that it's best to use experimental products in experiments rather than going off on the side. That said, I do provide this product to patients who really want it. It is not something where I think the safety profile is such that it's reasonable, again, for people who are highly motivated and who cannot be in a trial to test it out, because it is, again, you can do it legally, and again, as long as it's supervised, I think it's not particularly dangerous. The safety profile is good up to at least about 100 milligrams a day, but we don't know the optimal dose in ALS, and we're testing somewhere between 60 and 100 in this study. The approved drug dose is 20. So kind of finding a dose there, and again, one size fits all. So kind of figuring out what a dose that's easily tolerable if you're going to do it, that doesn't push you into tolerability issues. A lot of our other products in ALS also have GI tolerability issues. So kind of we're stacking a couple of drugs there together that has a rather similar tolerability profile. So again, figuring out what kind of works for a person there. The drug is not very expensive, but I think that in the US, importing it from Japan ends up costing something like $500 a month or so. So it's not cheap either. I mean, it is, but again, self-experimentation, I think I wouldn't recommend it, but I participate when somebody really wants it. That's kind of how I feel about it. I try to follow outcome measures and really listen to the person who starts the product. If somebody starts a non-approved therapy and feels worse with it, well, then I don't think one should persist for any length of time. If one starts something a bit off-label and really feels that it is helpful, then trying to kind of corroborate that with some objective evidence. Today, since we have neurofilament light commercially available in the US, that tends to be a marker that I use, but also look at functional rating scale, vital capacity, ALSA-Q5, those are others. We do many other projects here. Again, we are probably best known for the C9 discovery, and many of the pathobiology programs regarding C9 ALS and how that, but we have a few other projects that are ongoing. We work with many patients, both from the US and abroad, and the families of our patients. We have a large tissue repository, a clinical team, a group of other physicians who also help out and see ALS patients. We have a clinical research team, and these are all leaders of basic science groups that each have their own laboratories that are associated with us. And I also should acknowledge our funding sources. Thank you. This is a picture painted by one of our patients here who is quite public. She does paint with her feet to achieve these kind of beautiful pictures. And thank you.
That's great. Excellent. Thank you, Dr. Oscarsson. You can stop sharing your screen now. So thank you very much for that excellent presentation. Very interesting and very exciting indeed. So my first question is kind of a typical question. You are already familiar with the anxiety that comes from the ALS community. So it's a pretty simple question. When can we expect next results for this Combat ALS trial? And are these next results likely to be definite or probably preliminary?
So we have an interim analysis coming late this year, we expect. It may end up being early next year, but somewhere around that time, we should have an interim analysis. That will be a go, no-go decision. And again, if we get a no-go decision, is it truly definitive? I guess we wouldn't. There's sort of that question, so what's the minimal meaningful difference for a product to be meaningful? And That's a difficult question, ALS, and particularly if we're thinking that a product has a different effect in different patients. Again, this responder analysis that was done on the earlier study hint at that. Is that really true or not? It's hard to know. But yes, it's possible that we would miss an effect in a smaller subset of patients at that point. When would we really be done with this project? Our current enrollment is picking up. We picked up speed enrollments since this spring, but it isn't the fastest enrolling study in the US. In Canada, it's doing pretty well.
Good. Okay. Thank you. And so in the last year or so, we've had two newly approved drugs in ALS, AMX and also tofersen for SOD1 patients, at least in North America. So what's your take on the current landscape of ALS research?
So there is a interest from pharmaceutical companies to get into the ALS space. And that is very encouraging. I think that speaks to that we have targets that they can drug. And then we have an infrastructure of trial sites and certainly a very engaged patient population where many people are willing to give up themselves as volunteers. And we have that structure built that lets us do trials effectively in ALS. So there's an unmet clinical need. There's a ability to do trials reasonably fast and reasonably well. So that I think that helps pharma stay interested. These approvals likewise are, other pharmaceutical companies look at this and see that the bar isn't set too high. Yeah, I think that many look at it and think that, yes, if they can do it, we can do it too, based on these numbers. And so it's not like the bar for success is set very high. So I think these factors will further drive keep pharmaceutical companies interested in ALS. I am very hopeful about that. And I think that they are the ones who need to take that final step. Yes, basic discovery, you look to academia for that, but then to actually apply it and get it out to people, that's what we use industry for.
Clear, thank you. And so in case Ibudilast is hopefully approved, how do you think it will combine with the other approved drugs given its target mechanism?
So again, it is in it of itself has potentially multiple mechanisms. I think that, again, on an FDA label, it would say mechanism unknown. Because there isn't one that's sort of clearly a standout, the effect of this product. We are studying it together with all the other products. So the studies that are ongoing, the combat study allows for riluzole. It does not demand it. We'd first started out that way, but we have relaxed that criteria. So you can be on riluzole, but you don't have to. You're allowed to be on edaravone. You're allowed to be on the Relyvrio, the TUDCA sodium phenylbutyrate combination. So it really is an experiment that we're doing on top of the others. So an effect that we saw would be an additive effect to the current standard of care. So yes, it would add well. If we see an effect there, it would be proven in that setting.
Great, thank you. You already anticipated this question in during your presentation, but I think it's important to highlight this. So we know that Ibudilast is currently available in Japan for a very different indication. So can you comment a little bit more on that, on how patients should deal with experimenting a drug that is already commercially available in the market, but for a very different disease?
Yes. So again, my advice is don't do it, but then I'm also telling you how to do it if you still want to. I think that looking at cost and again, making sure that trying to first start the currently approved products that are available to you. That's kind of where I would start. And then if in case you absolutely feel the need that you must test it, working with a neurologist and having your primary physician and internist kind of be aware of that you're going to be doing this. One of them will need to be prescribing the product, and then would also need to be sort of feel responsible for this and kind of keep an eye on you to work with you as you're testing this out. Having outcome measures collected in advance, so having a slope collected, that's generally easy to do. Normally takes a little time to acquire a product, and having collected outcome measures so you kind of have an idea about how your disease is doing, at least by the measures that we have today. I think starting pretty low with this product is still best. I mean, we start at 30 milligrams twice a day in our protocol, and that works pretty good. But for people who are using it off-label, I often even start lower. I mean, it's also a cost issue, and again, sort of since we don't know what the ideal dose is, maybe going a bit on the lower side. And then increasing it up to your target dose. I think often I put 60 milligrams as sort of my target dose, 30 milligrams twice a day for people. But again, we don't know. Somewhere between 20 and 100, I guess, is, I think that's the boundaries that we have. And again, keeping an eye on, there's not really any blood monitoring that I think is strongly indicated. We normally follow liver functions just because of riluzole. But having periodic liver monitoring and making sure that nothing strange is happening. Whenever you self-experiment, I think there's many of the people who do it tend to utilize many products. And then it gets really dicey to kind of sort out if something happens, if liver functions start going up or some blood counts start changing, which of these products is it that is to blame? But again, working with a team of a neurologist and an internist in that situation is normally the best way to go. Yeah, I think that's kind of the advice that I have. So again, and if you run into side effects with the tolerability, just kind of back dose down to somewhere where it is easily tolerable.
Good. Thank you for that. So finally, would you like to share us something about any other ALS study you're involved in that you find particularly exciting?
Well, so I mean, we still don't understand enough about ALS. And again, much of what we do is collection of samples and the study of different patient samples, both blood, spinal fluid, and nerve tissue, to understand what ALS is. And I think that while these projects have a longer horizon before they read out, I think that that's still probably where we should be, where I see the most potential. We might stumble upon something. We have the genetic forms of ALS now, and the list of genetic explanations for ALS is steadily growing. We're starting to be able to, we have the tofersen, we have other products in trials that are also striking at other genetic targets. We're kind of at the point where we can request and sort of petition for having gene-specific drugs created. And I think that is, I'm very intrigued by that, and I'm hopeful that we'll have drugs that end up really being truly individualized for people that work, that are potent. So we may understand ALS well enough for a portion of people, and I'd like to see us actually curing that smaller group of people while we're still searching for cures for the rest, and hopefully stabilizing people adequately as we're stacking up these weaker agents that we're have approved today.
Fantastic. Thank you so much, Dr. Oscarson. We really appreciate this very valuable educational contribution from your side. Congratulations on your work. And thank you for working on ALS. We must say that every day for all the experts like yourself that are really fighting to find a cure or maybe better treatments, more effective treatments for our patients. So we really appreciate that you have accepted to give this conference for our community in Argentina and in the rest of the Spanish-speaking countries around the world.
Thank you for having me. And yes, let's keep up the fight.
Sure, we will. And hopefully we will see you soon, maybe in Basel, Switzerland, by the end of this year.
I should be there, so yes.
Okay, we're looking forward to it. So once again, thank you very much, Dr. Oscarson, and see you next time.
Thank you.
Take care. Thank you so much.